Okay, so hello, I'm generic human being 6,143,918,033 here. I know that this has probably never been done before, where people do tutorials going through A-level papers. However, I thought I'd give it a spin of my own. So a little background, I'm a fourth year medical student at the time of making this, and I am an A-level biology, chemistry, physics, and maths tutor, and also a tutor for GCSE or any level for that matter from the beginning of time all the way until you're 18 years old. So I hope this helps. If it does, I'll carry on doing this, and yeah. Starting off, so I'm going to be going through the 2020 paper 1, which is all on the AS content, so all the stuff you'll learn in the first year of your A-levels. So, looking at the question, so we'll start off here. In general with biology, it's good to know what actually, what part of the spec it's actually testing you. Whether it be, this topic or that. So having a vague idea of where topics sit helps a lot. Figure 1 shows a cell of the lining of the ileum specialized for absorption of products of digestion. SGLT1 is a carrier protein, so this is a pretty interesting fact you should keep in mind, found in the cell surface membrane of this cell. It transports glucose and sodium ions into the cell, so you guys should be familiar with this mechanism. It's something you need to know. So the action of the carrier protein X is linked to a membrane-bound ATP hydrolase enzyme. So when you get a diagram like this, it's quite easy to just be thrown away by all the arrows and stuff. So it's helpful to just go through it one by one and see what's actually going on here. So you can see that sodium is moving out, potassium is moving in. This is your carrier protein X. And you can see that glucose is coming out here, but it's going in here along with the sodium. So the sodium's moving in down a concentration gradient and bringing the glucose along with it. For this to work, there has to actually be a concentration gradient to start with, which is why this sodium is being pumped out here and the potassium is being exchanged in its place. As the sodium moves out, the concentration of sodium in the cell drops, meaning that the concentration gradient from the lumen, which is where all the food that you've just scanned is, the concentration is higher here than it is inside the cell. So this facilitates the diffusion of sodium into the cell. As well as that, because this is a sodium glucose co-transporter, the glucose moves along with it. So now that we've made sense of what's going on here, it's now the next part is to actually tackle a question. So the action of the carrier protein is linked to a membrane-bound ATP hydrolase enzyme. Explain the function of ATP hydrolase. With any question like this, you should always have a model answer ready to go. ATP hydrolase, hydrolysis A's, enzyme that hydrolyzes ATP. So it hydrolyzes ATP into ADP plus inorganic phosphate, also known as PI, to release energy. You should always specify that it releases energy. The reason being is because you saying it hydrolyzes the ATP doesn't really tell the examiner why it's being done, if that makes sense. So for example, if you were to ask me, if I were to tell you a car has 4 wheels, it doesn't tell you that it's useful for driving, it just says that. So this would be your first mark. Now obviously, they wouldn't have asked you this question alone they wouldn't have asked this question and this diagram for just this point alone. You need to specify what the actual energy is used for in this case. And that's where your second mark comes in. So if we look at what's going on here, we need to look at why, if you know the mechanism of this co-transporter, then you will know that this sodium-potassium pump uses active transport. So the energy is used in active transport. So in biology, a lot of mistakes that people make is that they'll say it hydrolyzes ATP, expecting that to be a mark in its own. And it generally can be, but most of the time isn't. It's always, you got to say a statement, therefore, the next thing that follows is usually 1 mark from what I've seen in my biology tutoring. I recommend just saying this by default in all questions where you're talking about hydrolyzing ATP. Right. So the movement of sodium out of the cell allows absorption of glucose into the cell lining the ileum. So this is what I just explained before. We're shoving all this sodium out, so the concentration of sodium inside the cell drops. So if the sodium inside the cell drops, then the sodium inside the lumen, the tube that, the digestive tract of all the stuff that you've just eaten, is going to be higher than inside the cell. So it's going to move down a concentration gradient, and it's going to bring the glucose with it. So now we need to put that into the words that the examiner wants, basically. So what does it do? So the movement of Na plus, do not write sodium alone, it has to be sodium ions or Na plus, allows the concentration to stay lower inside the cell than the ileum lumen, or you could just say it maintains a concentration gradient. There's also that. Then what? So you've said that first part of the explanation. The next part is explaining why the glucose comes in. So sodium ions move in down this concentration gradient that you've just made. And write conc for short. You don't do that in your exam. Down this concentration gradient via facilitated diffusion. You need to specify what mechanism it is. Bringing glucose with it. So another exam technique thing you should always be wary of is that if you're ever talking about the movement of substances, you always need to clarify, is it diffusion? Is it active transport? Is it is it simple diffusion? Is it facilitated diffusion? Is it active transport? Is it osmosis? Is it from a low concentration to a high concentration to a low concentration, or a high water potential to a lower water potential? You always, 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 always must include that, or you'll just lose marks. And even if you don't get, even if it's not something that the mark scheme wants, there's no harm in actually writing it. Next, describe and explain. So describe and explain two features. Describe and explain two features you'd expect to find in a cell specialized for absorption. So the thing that people slip up on here is that they say the feature and then they don't explain. So describing is saying what the actual feature is. Explaining is why this is good. So if we're looking at cells specialized for absorption, we're going to expect that they have microvilli. So you would say the cells have microvilli. One other way you might see this question is structure and function. Structure is the equivalent of describe here, function is the equivalent of explain. So you've said the structure here, or you've described it. Now you need to say what the function is or explain. So why is this good? So cells have microvilli, so there is an increased surface area for absorption. That's all 1 mark. You cannot, saying cells have microvilli is the same as again saying a car has 4 wheels. It doesn't tell me what the 4 wheels serve, the purpose of the 4 wheels is. So saying cars have 4 wheels, therefore you can drive it and get to places, is more useful than just saying cars have 4 wheels. In the same way here, it's more useful to say cells have microvilli, so there's an increased absorption, surface area for absorption. Next, another point, so you can think about other things that they might have. So this question here, sometimes you might forget, but you can use pointers from the question. You know that this is involving ATP and energy and active transport. So it's likely that there's going to be a lot of ATP involved. What makes ATP? Mitochondria. So therefore, these cells that probably, cells typically that have a lot of active transport going on or a lot of energy requirements are probably going to have a lot of mitochondria in them so they can provide energy. It would be daft of them not to. So you would say large numbers of mitochondria. There's your describe. So more ATP is made to provide energy from respiration, and that will guarantee you your marks. Okay, so make sure you're aware of that. When questions are talking about cell specialization in this context, they're talking about features, because if you remember, plant and animal cells, or any type of cell on the planet, does not have every single organelle in the exact proportions you see in diagrams. Some cells look different in shape. Some cells have more of this and more of that. Right. So this is a question that everyone kind of died on in this one, and it's really not that tricky. So it says figure 2 is a diagram of one SGLT1 carrier protein. And it's told you earlier on in the question that it's embedded in the plasma cell surface membrane. All this is asking you to do is to show a demonstration of the fluid mosaic model, basically an understanding of it. So your fluid mosaic model is that you have these phospholipid bilayer here, and then you've got these proteins embedded in between them. So in this diagram, you see the hydrophilic regions here, hydrophobic regions here. This whole thing is the protein. So it's embedded within the phospholipid bilayer next to each other. So you're going to have your heads out like that and your tails coming in like this. Like so. All it's asking is to do that. A lot of people looked at this and kind of panicked and knew that they needed to do this but didn't know where to put it. Remember from your your textbook diagrams and stuff, this is what it would appear like. So next, do not draw more than 8. Okay, fine. Oh, whoops. Okay, fine. I've just read that by mistake. Right, fine. So figure 2 shows the SGLT1 polypeptide with NH2 at one end and COOH at the other end. Okay, right. So the general structure of an amino acid is you've got a hydrogen here, a carbon here, an NH2 here, and then an HOOC here, and then a variable R group. That changes depending on what amino acid you have. So you need to appreciate how the you need to basically demonstrate how these join together every single time, so that there's always a COOH on one end and an H2 on the other. So I've made this diagram a bit too big, haven't I? HOOC, C, NH2, R, H, then HOOC, C, NH2, H, R. So what comes out of this is, I'm actually going to redraw this a little bit slightly differently, is that. So an H goes from here, and then an OH goes from here, and you get this. HOOC, C, N, H, R, H, C, and it's a double bond O, C, N, H2, R, H. That's how it forms. This is a peptide bond. Now, what do we actually write in this question? So, you can say that one NH2 group bonds to the COOH group of another amino acid, forming a peptide bond is a smart way of saying this. You should always, I'll get onto this one sec. Forming a peptide bond. Whenever you talk about bonds being made, you must always clarify what bond is being made. If two amino acids join together, you are making a peptide bond. If two, glucose molecules are joining together, you're making a glycosidic bond. You need to clarify, or you will not get the marks. So there's that. And then obviously here you can express with the diagram, but you need to basically articulate that every single time this happens, one end is going to have this, one end is going to have that. So how would I phrase this? There will always be a free NH2 at one end and a free COOH at the other end of a polypeptide chain. That's what I would write. Okay, so there's that. Question 2 then. So, question 2. So it says, to study lipid digestion, a scientist placed a tube into the gut of a 20-year-old man. The end of the tube passed through the stomach but did not reach as far as the ileum. The stomach fed a man a meal containing triglycerides through the tube. The scientist also used the tube to remove samples of the man's gut intervals after the meal. They recorded the type of lipid found in the sample. Some of the results are shown in this table. So, what we got? We got sample A here and B. Time of collection after meal, 45-75 minutes. Concentration of fatty acids. After 30 minutes in this second sample, you can see it's gone up, and the triglycerides has dropped. So use your knowledge of lipid digestion to explain the results in this. So what's happened is this person's effectively eaten something, this person's effectively eaten triglycerides. So it's just asking you to, and they're just measuring the sort of results that come from it. So you can see that the concentration of fatty acids increases and the triglycerides decreases. So this person is digesting these triglycerides. And what do triglycerides make when they are digested? Fatty acids. So that's what it's trying to articulate. So you need to explain why this is happening. So people are going to say, like, the common thing that I see is that people will say, oh yeah, concentration of triglycerides decreases from 0.6 to 0, and then the concentration of fatty acids increases. That's again the equivalent of saying a car has 4 wheels and not explaining why. And that's a common mistake. You need to interpret and tell the examiner what's going on. So here, I'm going to start the triglycerides. The triglycerides, pardon my handwriting, decrease because of what? Because they're being digested, aren't they? What digests triglycerides? Lipase. Because of digestion by lipase. Now, a lot of people will then go on and say, oh, okay, then that means fatty acids increases because of the lipase. They're both usually the same point. So if you were to say triglycerides decrease because of the digestion of lipase and the fatty acids increase because of lipase, they're the same point, because I mean, if you break this down, you're going to make that, so it doesn't really count as a separate mark. You need to now clarify why, this lipase leads to the breakdown of this. And I'm going to give you a tip on this after. So you would say that the lipase hydrolyzes ester bonds between fatty acids and glycerol. And the mark scheme shows that this is a mark and this is a mark. Whenever you talk about making a molecule, so you're making it, like a polymer or whatever. In biology, always say what the monomer is, the type of bond formed, and how the bond was made, which in most cases is condensation. Okay, so if you were to talk about, for example, making a dipeptide, you would have amino acids, that's your monomer, forming a peptide bond via condensation reactions in which a molecule of water is made. Same principle goes for breaking a molecule. Every single time you say these exact same 3 things, but actually you kind of go backwards. You say the bond broken, how the bond is broken, so that would be hydrolysis in most cases, and the product's made. So if I'm breaking said dipeptide down, I'm breaking a peptide bond via hydrolysis, producing 2 amino acids. If you say this in your answers, you first of all can get all 3 marks by saying a very short sentence if you can combine all of them together, but you're guaranteeing yourself those marks every single time. So do not do this. Always do this. Right. Let's see what's the next question then. So, after collecting the samples, the scientist immediately heats them to 70 degrees for 10 minutes. Let's see why this is going on. So this experiment involves the use of lipase, doesn't it? So let's see. So it doesn't really say in the question. If you can't establish that lipase is the result of what's going on here, then you're kind of screwed for this question. If you take a sample out and the lipase is still working, the numbers are just going to keep decreasing. So we need to stop lipase working. So the answer here would be to denature lipase, so no further hydrolysis takes place. Now, a lot of people will say break down or whatever. Some mark schemes accept it, some don't. We're in A level now. You've got to be saying hydrolysis instead. Now, describe the role of mycelials in the absorption of fats into the cells lining the ileum. So this is something that people also do not like a lot of the time. So I'm going to just draw a quick diagram to explain what's going on. So when you break down a lipid, you make a monoglyceride and your 2 fatty acids like so. Okay, so this is the ileum, and this is your gut the inside where all the digestions happen. These are the just freshly broken down lipids. So we've broken down a lipid into a monoglyceride and 2 fatty acids. The problem is because they're non-polar, they can't really move around, and they just kind of slosh about here until they get here. So we need something to help facilitate their movement out of the gut into the ileum cells and then into the body. So the thing with them being, because they're non-polar and because they are lipid soluble, they can freely just move through, but if we were to expect it to just happen on its own, it'd be very slow. So what happens is your body releases the bile salts, which I'm going to show as these green circles here, like so. So it releases the bile salts, and what happens is the bile salts associate with, so they kind of like stick onto the monoglycerides and the fatty acids and help them all move together. They carry them across like that, they kind of drag them across. So the green bile salts move and they drag the fatty acids across. Eventually, they can just pass through freely, and these stay behind and they go and just carry on, like so. When the fatty acids and the bile salts are associated with each other, when they're like stuck together kinda, they're called myceles. So the myceles, again, I'll just reiterate, drag the fatty acids along. They just pull them along like this. The bile salts themselves can't move through, so the fatty acids and the glycerol and whatever moves through, but the myceles dissociate and the bile salts stay behind. That's how it works. Then this goes into the cell, to the smooth endoplasmic reticulum, blah blah blah, wherever it needs to go and gets used. So now to answer this question here, how would we write this in a more effective way? You can say that bile salts. Bile salts associate. That's a nice way of saying mixes with, but kind of isn't, like, it's not really bonding with. Associate with the monoglycerides and fatty acids, forming myceles. They bring the fatty acids to the cell lining of the ileum. And remember what I said before is whenever things move in biology, like substances, you need to clarify how. Now, because fatty acids are lipid soluble, it's in the name, fatty acids, they can just be absorbed by diffusion. So you would say the fatty acids and the monoglyceride are, I'm going to write just fatty acids because I'm lazy, but yeah, are absorbed by diffusion. And that will guarantee you every single mark. I hope that makes sense. Right. So now, this question, I love this one because everyone shits themselves on this one too. Pardon my language. So we've got, so it says figure 3 shows the pressure and blood flow during the cardiac cycle in a dog. So you've got the pressure in the left ventricle, you've got the pressure in the aorta near the heart, and then we've got rate of blood flow in the heart. Now, one thing to appreciate here is that pressure and volume are inversely proportional. So as pressure increases, volume will have to decrease, or the other way around in general. So if I have a box here of this volume and I've got like 3 squares in, if I decrease the box volume, then the 3 squares are much closer together. So the pressure is higher, but the volume's decreased. So this is an important thing to appreciate. The pressure in the left ventricle is higher, then what we can assume is that the volume has decreased. Why would the volume of the left ventricle decrease? It's because it has contracted. If it's contracted, the volume is smaller, the blood is being squished together and forced out, so the pressure increases. So now, at P, the pressure in the left ventricle is increasing. At P, on figure 3, the pressure in the left ventricle is increasing. At this time, the rate of blood flow has not yet started to increase in the aorta. Use figure 3 to explain why. So we're looking at P here. If we look at P, these graphs all correlate with the same time scale as well. This is something that other people messed up on, is that they looked at this graph and thought, oh shit, there's 3 graphs here, I'm so scared. When you see graphs that look unfamiliar, just make sense of what the axes are talking about. So we have 3 different quantities being measured here, but they all use the same time. So at this point, P here are about what, like 0.38 seconds. You've got P here. So at P, the pressure in the left ventricle is increasing at this time. The aorta, the rate of blood flow is not increased in the aorta. Why is that? So you guys have to remember your sort of biology facts here. So when the ventricle contracts, if it wants to force, It wants to force blood through the aorta. And what stops the backflow of blood again? The aortic or semilunar valves, and they only open when the pressure behind them is high enough. The pressure is high enough if they open like that and the blood gets past, but they don't open until the pressure is high enough. So it says here the pressure is increasing at P, but it's probably not reached the high enough for the actual, valve to open here. So you would say here that you can say aortic or semilunar, either's fine. The semilunar valve has not yet opened. That's one mark. We need to clarify why. The pressure behind valves has to be higher. So if the pressure behind the valve is higher, then it will open. So what's behind the semilunar or aortic valve? The left ventricle. What's after it? The aorta. So you need to say that the pressure in the, in the left ventricle has to be lower than in the aorta, doesn't it? Or else it wouldn't work. So you would have to say the pressure behind the valve, which is in the ventricle. So the pressure in the ventricle is less than the aorta. And there's that one. Right, next. So, let me move this. Okay, so let me move this figure down here so I can see it. The joys of using Microsoft Whiteboard. I hate it. Okay, so at Q in figure 3, there's a small increase in pressure and in the rate of blood flow in the aorta. Explain why this happens and its importance. So, we're looking at figure 3, and we've got, so the rate of blood flow in the aorta near the heart, so the aorta, the rate's high, and then it kind of decreases, then blips a little bit, and then comes back up again. So, why might that be the case? Well, so what's happening here is that your blood is being forced through this aorta, and then it just sort of blips like this. The reason being is because it's expanded and then it's recoiled a bit, and that increases the pressure, increasing the blood flow. And that's actually how arteries in the body can help maintain the blood pressure. So what's going on here is that the aorta wall elastically recoils. It like squishes back in. I don't want to say squishes back in. It comes back in, and that obviously maintains blood pressure. Because remember, I'll write this down first. Maintains blood pressure. Because remember that as the volume decreases, the pressure increases. So if it's recoiling and it's tightening around the blood that's in it, it's going to increase the pressure, allowing blood to continue flowing. It's going to continue flowing at a faster rate. An example is if I had a hose pipe and I put my finger over here, then the water's going to spray out faster. That's because there's decreased volume and increased pressure. Whereas if I just let it like this, then it comes out a little slower. Right. Now, a student correctly plots the right ventricle pressure on the same grid as the left ventricle in figure 3. Describe one way in which the curve would be similar and one way it would be different. So they've put the right ventricle pressure on the grid of the left ventricle pressure in figure 3. So okay, so what would the similarity be? Well, remember in your heart contractions, you've got your atria contracting and then your ventricles contracting. They contract at the same time. So imagine I draw this schematic here of the atria and the ventricles. These two contract first, then these two. Because the reason is you want to get the blood in here first, and then you want to get the blood out here like this. So when you contract the first time around, the atria, they squeeze all the blood into the ventricles, and then the ventricles contract and then force it out into the body. They have to have that sort of the pattern. Reason being is because if all four of them contract at the same time, the blood's just not going to go anywhere. And also you don't want it so that one contracts and the other one kind of doesn't, and then whatever, like they contract at the same time. So if the ventricles contract at the same time, then the patterns are effectively going to be the same. So there's going to be a same pattern, really. The shape is going to be the same. The timing is also pretty much going to be the same. What's the difference? So now we need to compare the left and right ventricle, and everyone, fingers crossed, should know this, is that the left ventricle is fatter. Well, not fatter. The left ventricle is like chunky. It's got like mad muscle there. So obviously it contracts with more force. It increases the blood pressure. It has a higher blood pressure when it does that. So the contraction of the right ventricle must have a lower pressure. And it only says describe, so you can get away with saying lower pressure. If it said describe and explain, you would have to say lower pressure because the right ventricle doesn't have as much muscle in its wall. And the same for this. But because it only says describe, you can get away with just doing that alone. So now, Use information for figure 3 to calculate the heart rate. So this is something that everyone screwed up on as well. All you need to do is you need to find a point and then work out when it repeats, kind of like waves in physics if you've done physics in GCSE. So I'm going to pick this point here and then look for where it repeats again here. So that's what, 0.38 seconds, and then this is what, 0.6, 0.7, so 0.78. All right, so you want to work out the time difference, because that will tell you the time of, that will tell you the time of one cycle, one beat. So time for one cycle equals whatever those two subtracted are, which conveniently works out to be 0.4. And this is in seconds, 0.4 seconds. So if one beat is 0.4 seconds, then, you know, to go to 60 in 60 seconds, how many beats can we fit in? Well, you just do 60 divided by the 0.4. So 60 over 0.4 gives you, if I do this now on my calculator, 150. Now, this might not be what the mark scheme says. It's probably because of the scale, blah, 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 whatever I read, but the method still applies. And also because I read the grid like really rubbishly, but you'd still do that. Right. So now, moving on. Okay, so anthocyanins are colored pigments found in the cell Anthocyanins are colored pigments found in the cell vacuole of some plant cells. Anthocyanins cannot move across undamaged cell membranes. So this question presumably is going to start going on about cell membranes. A student investigated how to extract anthocyanins from blueberries. They mixed 10 grams of crushed fresh blueberries with 100 centimeters cubed of extraction solvent for an hour. They investigated 3 different extraction solvents. So what I'm guessing is they've got 3 different tubes here of stuff, and they put these crushed berries in, and they're looking at how much stuff gets released. So when making up the extraction solvent, they used a volume ratio of 70 to 30 to 1 of ethanol water to acid. Tick one box that shows the most appropriate volumes to make up 100 centimeters cubed of this extraction solvent. So you just need to divide 100 into this proportion here. So when working with ratios, what you want to do is you want to work out the total shares, if that makes sense. So you're sharing 70 to 30 to 1. So the total shares is going to be 70 plus 30 plus 1, which is 101. Now you want to work out one share of the total. So that's going to be 100 divided by 101, which is 100 divided by 101, which is what, like 0.99 blah blah blah. Just save that in your calculator. Then you want to work out the then what you want to do is you want to work out each of these shares basically. So if one share is 0.99 god knows what, then 70 shares is going to equal 70 times that answer, which is 69.3. Then 30 shares is going to equal 30 lots of whatever that thing is you worked out, which is 29.7, and then one share we just worked out 0.99. So now we need to figure out which one of these fits that basically, which is this box here, 69.3, 29.7, and that. So that's how you do ratios in a nutshell, because everyone looks at ratios and like, oh my god, I can't do it. Just do this and you'll be fine every single time. Right. So the student kept constant the mass of the fresh blueberries, the volume of extraction solvent, and the time for the mixture to stand. What else should be kept constant? Well, we're looking at things being moved around and, you know, things being released, like, you know, these anthocyanin pigments. So a good bet really is temperature. You want to keep temperature constant because you also don't want to damage the cell membrane by increasing it too much. And we need to think about other things. What else can affect the amount of sort of pigment being released from damaged cells? Well, it's also how long you're crushing it for, how you're, you know, what kind of blueberries are you using? Are you using really old rubbish ones or really good ones? And also, you want to control the concentrations of the ethanol, water, and acid. If I use one concentration here and then a different one in F, then it's just not going to be a valid experiment because, you know, I can't definitively say that is because of the ethanol. Because if I do a little bit in one experiment and then loads in the other, I can't tell if the change was because something else was there or because I increased the concentration. So I'm going to write temperature and concentration of ethanol and acid. But there's loads of things you can say here. Right, next. So, right, question, where's it gone? Here we go. So here we go. So we got, after 1 hour, the student filtered the samples. They placed the filter in a colorimeter and measured the light absorbance. The results are shown as follows. So, okay, we got E, F, G. I'm just going to write down what E and F and G are. So E is ethanol plus acid plus water. F, I think, was just ethanol alone. Yeah, well, I'm sorry, ethanol and water. So ethanol plus water, and then G was just water alone. So we need to, we need to explain what's going on here. So if the absorbance is higher, then we can, if the absorbance is higher, that must mean that more anthocyanin has been released. So it says we need to explain the results. We need to explain what the different, why the absorbance is as so. So higher absorbance means, I'm gonna write arrows, but you write this out in full. Anthocyanin release. So we've got that part. Now we need to compare all of these and explain why, each one has the results they have. So why is ethanol and HCl going to have the highest amount of anthocyanin release? Well, it must mean that there's more damage occurring, because of the acid presence. So, excuse me. We need to explain why this one's so high. Well, obviously the presence of acid means there's more, it's going to damage the cell membrane, so more is released. So we need to write that down as well. Is that more membrane damage leads to, more anthocyanin release. Release. Yeah. So now we need to compare all of them. So E is greater than F because the acid is present. So because the acid denatures proteins, we all can get that one. Now the bit that everyone's screwed up on next when I've seen people do this is that they completely just dismiss the fact that F is significantly higher than G. So F is higher than G, but we need to say why. Well, we're comparing ethanol and water. What does ethanol do? Well, ethanol dissolves lipids, and that includes phospholipids as well. So, F and G, because ethanol dissolves phospholipids. That's something that people forget. Remember your little lipid test where you add ethanol and some water and you shake it and you get a milky cloudy white, emulsion? That's, because ethanol dissolves lipids, including phospholipids as well. Next. So a different student did this investigation, but they did not have a colorimeter. So they need to prepare color standards and use them to give data for the total anthocyanin extract. So we're in a bit of a tricky situation here because we don't have a colorimeter. We can't measure numbers. So basically what they're doing is the only option we have is to get these tubes and then measure how dark they are. So imagine that's kind of dark. This is like a little bit dark, and this is like hella dark. That's the only option we have. We don't have any other way of, measuring how dark. So what we can do is we can use known concentrations of anthocyanin and then prepare a dilution series. Because let me write this down and I'll explain. The reason we'll do that is because if I have this, let's say I have this hella concentrated stuff here, and then I dilute it into, this, and then this, and then this, and then this, imagining that they're getting darker as we go along, obviously. If I have a test sample and I want to compare it and I see it's this, it might, and this is unknown, then I can say that this concentration matches this one that I know of. So let's say, I don't know, I'm making this up. Let's say this is 1 mole per decimeter cubed in its concentration. If you gave me this unknown sample, I could look at this one and be like, oh, okay, yours probably has a concentration of 1 mole per decimeter cubed. That's how this experiment works. Obviously, this is not perfect. And the only way to really theoretically get this to work well is to have an infinitely big series of dilutions, but we don't have a colorimeter, so this is the only option we have. So yeah, you would compare the unknown sample, the test sample. Oops, test sample with what we call with the standard solutions or the color standards of known concentration to determine the concentration, basically. Yeah. Right. So there's a blank page. Next. Okay, So again, like I said before, whenever we're making anything joined together, you always need to say the monomer, the bond formed, and the way it's happened. So I'm gonna write this down. I'm gonna get the mark scheme out and show you what it says. So DNA polymerase, what does it do? It joins DNA nucleotides. Those are the monomers. Whoops, via condensation reactions, because that's the mechanism by which it happens. Reactions to form, and the type of bond made, phosphodiester bonds. And this is me winging it here. And let me get the mark scheme out now. Hold on a second. Question 5.1. And I'll get this here. And would you look at that? Wow, exactly as I told you. It says joins adjacent, it's in brackets, you don't have to necessarily say that. And remember, in mark schemes, this semicolon separates a marking point. So, I don't know why it's tilted like that, sorry guys. It joins adjacent nucleotides, it catalyzes condensation reactions, forming phosphodiester bonds. Even though I only needed 2 marks, and within saying just 3 lines, you know, I've finessed all of the marks. So there's that. Right. So I'm going to move this down here to move on to the next part. So, figure 5 shows the percentage of rat cells undergoing DNA replication. Some cells contained a protein called cyclin D, and figure 5 shows the percentage of rat cells undergoing DNA replication. Some cells contained a protein called cyclin D, and some did not contain cyclin D. They were all in early interphase at time 0. So, look at the key and make sense. We've got percentage of cells undergoing DNA replication. We've got the time here, and then this is cyclin D. This is without cyclin D. So it took 25%. Sorry, it took less time for 25% of cells with cyclin D to undergo DNA replication than 25 without. Use figure 5 to calculate this percentage time. This time difference is a percentage decrease and show you're working. Right. So we need to find the 25% mark, which is here, 25. Move across until you hit the line, and then work your way down like that. So that gives us what, 11 here. Then you continue going across until you hit the other line. And that gives us 15, 16, 17.5. Okay, so to work out the percentage difference, it's always the second value minus the first over the first. So the first in this case is the one without cyclin D, so 17.5. So it's going to be 17.5 minus the second, which is with cyclin D, minus 11 or 11.5 or whatever you end up reading off that grid, over 17.5 times by 100, and that gives us 37%. Now, it's probably not going to match what the mark scheme says because I've read the scale like really badly, but the principle applies. All right. So, next. Cyclin D stimulates the phosphorylation of DNA polymerase, which activates DNA polymerase. Describe how an enzyme can be phosphorylated. So you're basically, phosphorylation by definition is sticking a phosphate on. Where do phosphates come from? You know, you guys should know, from ATP being hydrolyzed. So phosphates, or phosphate ions, are released from the hydrolysis of ATP into ADP and PI. And then the phosphate ion is attached to the enzyme. That's pretty much it. Next, so some tumor cells contain higher than normal concentrations of cyclin D. Use figure 5 to suggest why a higher than normal concentration of cyclin D can lead to a tumor. So we can see that with cyclin D, the percentage of cells undergoing DNA replication shoots up much quicker than over here. So what we can appreciate here is that cells begin DNA replication earlier, so they must have been starting mitosis earlier. So cells replicate DNA sooner and start mitosis sooner. So this can lead, and with any question to do with tumors, always just say it can lead to uncontrolled mitosis or cell division, leading to excess abnormal cells. That's anything to do with describing a tumor. If you want to describe a cancer, it would be uncontrolled mitosis or cell division leading to excess of normal cells which can invade and destroy surrounding tissue. That's what a cancer is. Next, particulate matter is solid particles and liquid particles suspended in air. Polluted air contains more particulate matter than clean air. A high concentration of particulate matter results in the death of some alveolar epithelium cells. If alveolar epithelial cells die inside a human body, they're replaced by non-specialized thickened tissue. Explain why this is the case. Okay, let's say we have some alveoli here. This is a very, very primitive diagram. Alright, this is a very, very primitive diagram. And imagine now we've got some thickened, scarred rubbish tissue here. You can already see that the surface area is a lot lower because you've got all of these little points here all the way around them for the diffusion to happen. You can also see that because it's thicker here, the diffusion pathway is longer. So what you want to say here is that there's reduced surface area. I'm going to write SA for short. There's an increased diffusion pathway or distance leading to reduced gas exchange. Exchange rate. You can't just say it reduces gas exchange. You need to appreciate that it reduces the rate of gas exchange because it's saying it's slowing it down. That's the explanation. Overall, the end result, is that there is reduced gas exchange. We need to explain why there's reduced gas exchange. The reduced surface area and the increased diffusion pathway means that the rate is slower, meaning there's less gas exchange overall, as in the speed of it. Scientists grew alveolar epithelium cells and exposed the epithelium cells to different concentrations of particulate matter, and they calculated the percentage of these alveolar epithelial cells that died after 24 hours of exposure to particulate matter, and their results are shown in this figure. So it's asking you, do the data show a linear relationship between concentration of particulate matter and the percentage of dead cells? Use suitable calculations to justify your answer. So this question here is probably one of the most tricky questions I've ever seen in a biology paper, and honestly, if you can't get calculation questions, don't stress it out. They're only worth a lot of work for 2 marks. Right, okay, so what I'm going to do is I'm going to work out the difference between each jump and then divide it by the concentration here, and that'll give us by what factor these climb up in. If it's linear, the factor should be the same. So this scale goes up in 1.5, so this is going to be 6, this is going to be 15%, and this is going to be 42%. So I'm going to work out the jump between here, so that's plus 9, and then jump here, which is plus 27. So I'm going to work out the factors here. So jump 1 is 9 over 5, which is 1.8, and this is in percent per microgram per centimeter cubed. Then jump 2 is going to equal 42 minus 15, which is 27, it's 27, over 95, which is 100 minus 5, and that gives us 28, sorry, not 28, 0.28. This is percent per microgram centimeter cubed. You can see that they're not the same, therefore, no. This is a very hard question. I wouldn't stress out about not getting it. It's only 2 marks for all of that work. Right. So, alpha-gal is a disaccharide found in red meat. It's made from 2 galactose molecules with this formula. Give the chemical formula for disaccharides. So if it's a disaccharide, you're putting 2 of these together. So you're going to do C6H12O6. You can times it all by 2. But remember, because it's a condensation reaction, you're leaving out water's being released. So you're minusing an H2O. So you're going to be left with C12H, and this would be 24, but you're minusing 2. So H22, O times 2, but you're taking 1 away. So that's going to be O11. So C12H22O11. And it says, describe how it is formed. So as with always, write down how it's made and what bond is made. So this is a condensation reaction forming a glycosidic bond. There you go. Okay, so some people eat red meat for many years without having any reaction, then have an allergic reaction to alpha-gal in red meat. An allergic reaction is caused by immune response. Draw a labeled diagram of an antibody and identify the specific alpha-gal binding site. So you will have your Y-shaped sort of polypeptide here. Well, let me make it a bit more clear actually. So you'll have your Y-shaped sort of chain here. So me and my beautiful artwork. You'll have your disulfide bridge here, and a disulfide bridge here. And then you've got your, you need to now basically label all of this. So this is a light chain. This is a heavy chain. That's a disulfide bridge. Ignore my handwriting, that's absolute disgust. This is the constant region. And then this is your alpha-gal binding site. Your variable region. And also that one as well there. You can make a NISA diagram of that. Whatever, right? As that. So, a tick is a small animal that bites humans and feeds on their blood. This results in proteins from the tick's saliva entering the human body. Scientists have suggested one hypothesis for the allergic reaction of alpha-gal in red meat. They think that an earlier immune response to a tick bite can cause a person to have an allergic reaction to alpha-gal in red meat. Suggest how one antibody can be specific to both of those. So I'm going to zoom in on that sort of the variable site, alpha-gal binding site, whatever, blah, 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 right? So let's imagine that there's this shape. Now this is completely made up just for the sake of simplifying this. Antibodies work because they are specific to a certain antigen. So an antigen is something that triggers the immune response. So if alpha-gal proteins are shaped like this, and maybe meat stuff, well, whatever, red meat has a similar shape like, like this or something, then what can happen is that if it's complementary to the antibody, the alpha-gal binds here, and then that other thing that I drew could also bind here as well. Like so. And that leads to the immune response. The reason being is because they're similar in shape, so they're both complementary to the antibody. So we need to just write this down basically, so into a way that the mark scheme would want you to say. So you would say that the alpha-gal antigen, proteins, and red meat proteins have a similar shape, so the antibody is complementary, this is a key word, to both the alpha-gal and the red meat. That's why it leads to the immune response, because it doesn't know how to tell them apart. If it fits, it sits. Oh, the figure's been removed. No! Alright, soz mates. I'll have a look at what this could be. Scientists took blood samples from one man over several weeks and measured the concentration of antibody in the man's blood. During this time, he had 2 tick bites and had an allergic reaction to alpha-gal in the red meat. Okay, so let's have a look. So I'm going to make up a table. So before tick bite 1, tick bite 2, amount of antibody. I'm going to use arrows. So to alpha-gal. So again, this might be like, I'm making this up really. So this question might have been something like, I don't know, before there was 0, then after a tick bite there was 1, and then after a second tick bite there was loads. And yeah. In this case, what you would say is something along the lines of, the first exposure leads to an increase in antibodies, and obviously as that immune response actually undergoes, some of the helper B and T cells, some of the helper T cells and the B cells go on to form memory cells, and once you're exposed to that thing again, they release more antibodies in a faster time frame and at a high, in a more sort of aggressive rate, leading to an increase. I don't know what this question would have asked. Sorry, lads, but I feel like it was something along those lines. Complete table 2 to show 3 differences between the DNA in the nucleus of a plant cell and DNA in a prokaryote. So, first of all, an easy one to really get with is that in a plant it's linear, because you have lines and they form a double helix, and then you have circular in prokaryotes. Prokaryotes are effectively just bacteria at level. What else can you say? Well, I mean, prokaryotes have plasmids. Plants do not have plasmids. What else? There's introns in the plant cell because they're eukaryotic, but there's no introns in prokaryotes. What else can you say? You can say there's histones, no histones, and you could say, what else is there? Longer and shorter, but longer and shorter is a bit of a risky bet. Right, so scientists investigated the genetic diversity between several species of sweet potato. They studied the non-coding multiple repeats of base sequences, defined non-coding base sequences, and described where non-coding multiple repeats are positioned within the genome. So basically, all it is it's DNA that does not code for protein. That's just the definition. And where would it go? It would be between genes. Okay, so a gene, remember, is a section of DNA that codes for a protein. So I like to use an example of cookbooks. So a gene, you know, your DNA would be like a big old cookbook, and in it are different recipes. A chocolate cake would be one gene, you know, a vanilla something or other sponge would be another. So imagine you're reading this chocolate cake recipe, right? And you scroll, you know, reading through whatever. Then all of a sudden, I'm talking about like my dad, you know, I don't know, like breaking my skateboard when driving off of the driveway, and then you're back to reading that. It kind of interrupts the flow. And to be fair, if you look at any damn recipe on the internet, that's what they do. They'll tell you some life story about God knows what before actually giving you the recipe. Anyway, we digress. Back to here. It wouldn't make sense to do that, would it? It would kind of just interrupt everything. So the non-coding regions are actually between the genes. So you could read the whole recipe and then read about how my dad broke my skateboard as he drove. I still don't forgive him for that. But yeah, that's basically what the principle is. So let's have a look here. So the percentage similarities in the non-coding multiple repeats of base sequences of 4 species of sweet potato are shown in this table. What do we got here? So obviously C and C are not going to compare because they'd be 100% similar in theory. C and L, they're quite similar at 53%. C and R aren't as similar. C and T are pretty highly similar, and so on. So you need to put a phylogenetic tree in place, basically. So because these have the most recent common ancestor, these two have to be the most related. So we need to find two things that are the most related. So if you look at the highest numbers in the table, you can see it's between C and T or T and C. So I'm going to write C and T here. The other two are going to be L and R. So we need to, you know, figure out which one comes next. Is it R or is it L? The way we do is we compare how similar they are to the T. So look at R and T and L and T. So R and T is 36%, L and T is 53%. Because these are branched off closer together, it's more like it's probably going to be the L that is here because they have to be more related, obviously, because these, you know, if you compare this box and this box, they're spread out further, basically. Yeah. This is going to be this. This L has to be more closely related to it because it branched off, you know, at a sooner point than R did. Right. Now, whoops. The scientists studied 5 individuals from each species. Within the 5 individuals of species T, so they've picked out 5 homies from species T and compared them, and they found that there's a percentage similarity of 66%. Surely, if they were the same species, they should have a percentage similarity of 100. So use table 3 to evaluate how this affects the validity of this phylogenetic tree. I'm not gonna lie, we're working on the assumption that, like, what this is saying is if we got T and compared it to T, this would be 66%. And I just said at the start, in theory, they should be 100. So this means this table doesn't really kind of work because we're working on the basis that C and C would have 100% match, you know, L and L and so on. So, you know, I wouldn't support it. Not, you know, it's not valid because there's because there is intra-specific variation, like a pretty chunky amount. And also, we're testing five samples, bro. So, you know, what is it? So small sample size. You know, you're only testing 5 individuals. Like that's rubbish. So there's a thing that I like to talk about when it comes to evaluating questions. I know we finished the paper, but this is something that I like to talk about. So a lot of the time people screw up on evaluate questions. When you see an evaluate question, what it's trying to get you to do is to say, is this correct? Is this not? And why? Why is that the case? So with all evaluate questions, what I recommend doing is, first of all, what's the sample size like? Is it big? Is it small? If I have 10 people in a room and I give 10 people chocolate and every person in that room shits themselves because I gave them chocolate, I can't then go and say that the entire population or anyone who eats it will shit themselves if they eat chocolate because I only tested it on 10 people and you and I both know that doesn't happen. Next thing to look at is what age of the people are involved. If the people are involved, age, sex, you know, health, and all of that kind of stuff. If I'm doing an experiment on people, I ideally want them to be the same age, because if I'm testing a drug on somebody who's a baby versus an old person, their body's going to process that drug like massively differently. Next, what you want to look at is timeline. Was this tested over a good amount of time or no? Then what you want to do is you want to look at things like, for example, was it tested, if it's a drug, has it been tested in the person's body? Has it been tested in a petri dish? Because it might be different. You will see that these questions usually have some sort of absolutely disastrous theory. So, for example, it will be something like a scientist tests a drug on the neck cancer cells in a test tube, and then a journalist reads this and thinks, all right, this drug can cure all cancer. They've just made a horrific assumption on the basis of, testing something in a test tube. First of all, they've tested it only on neck cells, and they've tested it in a test tube, so it might be it might not be the case on a real person. AQA specifically says that if a scientist does the experiment, you assume it's done correctly, so there were no errors in the methods. So moving back on, sorry, I digress. Other things is, have they done repeats? Have they, worked their averages? Have they done any statistical tests? If there are no statistical tests, you can't confirm something. It might have been due to chance. Are there, if there's standard deviations, are there any overlaps? Does it apply to all conditions, species, situations, like I said before with the cancer thing? Are there other factors that could come into play? Like, for example, maybe for example, if we did that experiment with the throat cancer cells, we sampled it from different patients, maybe because their health is different, they might be different. Are there any side effects as well if they're testing drugs? If we give a throat cancer drug to everybody, they might get side effects. Are there any financial things that could come into play? And also, is there subjectivity? So for example, if you do an experiment and you ask patients to tell you how dark, I don't know, a solution is, dark to somebody might be completely different to somebody else. If I asked you what your opinion of dark is, you might think black, I might think, dark gray. So those are the things that you need to ask. But as we go through more of these papers, we'll sort of establish ways and techniques of handling these. I hope that was helpful. I know I speak quite fast, but you could pause and do whatever, and feel free to comment any questions or anything you want to ask, and I'll do my best to answer them. Yeah. I also just want to put this out there as a disclaimer that I am in no way, shape, or form affiliated with AQA, nor do I intend to get any sort of monetary gain out of this. I'm simply just doing this because this is something that I wish I had at the time of doing my A-levels. Thank you.